All right, and our last lightning talk of the day is Mohsen Soltanifar. Woo!
Hi everyone, I am a statistician in my day job and teacher at academia in my weekend job. So this is my second R package that I developed and the first one in the pharmaceutical context. So I'm so excited about this package. Um, so it's called GenTwoArmsTrialSize. So this R package generalized established trial size package in the pharmaceutical context. So the outline is what's two-arm trial size, sample size calculations, the generalization perspective, this R package, and a working example in the American context. So why we do sample size in clinical trials? Because there are economic consideration. We want to cut the costs. Ethical consideration, we don't want to expose patients, extra patients to the new drug. And also scientific consideration, we don't want to end up to some futile results in terms of the estimates. So in this process, we have three items playing. One is the effect size, one is sample size, and one is power. In this R package, we are going to consider a sample size in terms of effect size and given power. So up to now, we have this situation. We don't have established statistical software that considers two key components in sample size calculations. One is loss of follow-up, and one is non-compliance. So in this R package, we are going to address these two key components. And other than that, we are going to generalize and uniformize what has been done in the previous R package and other statistical softwares, for example, inquiry, pass, and SAS. So these new R package which we developed generalize the one established trial size and also establish HMISC by one of the FDA statistician, Dr. Frank Harrell. So it has 20 scenarios in terms of endpoint, design, and hypothesis dense. So it begins with the two simple line comments to get installed, install package with dependency true, and also we call it by library function. It has four main functions. The first one, which deals with the continuous outcomes, get size min, has 10 parameters. The second one with binary outcomes, get size prob, has 11 parameters. The third one with the time to event outcomes, called get size TTE, has 12 parameters. And finally, the fourth, which deals with the ordinal outcomes, has 10 parameters, and its function is get size or. Now we show you one applied example of these R package in the United States context. So the sponsor of this clinical trial is Endologics, and they are going to compare two medical devices, the current and the new one that they developed in several American sites, 80 actually. So these medical devices are installed inside the veins, and they want to improve the flood of blood in the vein. Sometimes the veins of the patients get blocked or the blood flow gets slowed. So they install these medical devices to improve the blood flow and remove the blockage of those blood. So the sample size estimation information for this trial are in terms of the superiority hypothesis testing, 1-to-1 ratio, 80% power, and also the response of 86% in the new device versus 79% in the old device. So once we did that, we plot the two-dimensional plots of sample size in terms of the loss of follow-up and non-compliance. And here we noticed that for the chosen trial size 804, it matched to 1% non-compliance rate and 10% loss of follow-up. which makes sense here. Also, we changed the statistical software that calculates the sample size. Here we use SAS and nQuery. And here we notice that our results are pretty much with the SAS software. So we could generalize this R package in several directions, like multiple endpoints, multiple arms, standard effect size, adaptive designs, and also we can calculate power calculation in terms of given sample size and effect size. These are the tutorial and the source code if you'd like to go and find it. And thank you very much, and I appreciate your attention.